Alrighty. Hi, ladies and gentlemen. This is Trisha with Insectopia, and we have been celebrating Invertober or Inkvertober, and today we are going to be doing ink lines. So yahoo! We're gonna be celebrating Inktober together. And during Invertober, we are live streaming every day of the week. Monday to Friday, Saturday, and Sunday. Well, Monday to Friday, Monday to Saturday. Yeah, Monday to Saturday, we're doing 10 p.m. Eastern, and then Sundays, we're sticking it at 4 p.m. Except next Sunday, October 9th, because I'm going to be out of town at 4 p.m., but I will hopefully be home by 10. So we're going to be pushing next Sundays to 10 p.m. so that we don't miss a session. Yahoo! Welcome, Susan. I'm glad you are here and chatting in the box. If anyone else joins us, please go ahead and just give out a shout. Let us know you're here. Say hi. Join the conversation. All right. Let's see. And close and open. Aha! Alright, so today I thought that we would start off with the rough stink bug. This is Brachypnea quadripustulata. And this is a stink bug that is arboreal, meaning that it is found in forests, especially around trees. Alright. So it's arboreal, it's found in forests, especially high up in trees. And when we were looking at this specimen underneath a microscope, the texturing on the body was very bark or tree-like. And along the edges of the pronotum, that's this section here, this is our pronotum, along the edges of the pronotum here, there is a lot of kind of spines and spikes that I didn't take the time when I was originally sketching it and add individual. So when I'm doing this line here, I'm going to purposely make this one and this one kind of jagged, so that they're kind of textured like they would be in the specimen. Now, this is kind of the beginning or the first sketch in one of my sketchbooks, and I thought, because there's a series of them, this is our blister beetle this was that gray blister beetle that had the stripes along its back, and then not this one because that's the same species. I did him again. But I thought this one could be cool too. This is our soldier fly that we sketched, and we spent a lot of time making sure the wing venation was correct, and we talked about the decel, and we have kind of I'm pretty happy with this outline sketch, even though it's not super detailed and textured. I think that the line drawing, yeah, I thought that the line drawing was pretty impressive, so I want to turn this one into ink for sure. Yeah. And the scientific characteristic, so the characteristic that helps you identify stratiomyiids or soldier flies is this discal cell here, this D cell, and the fact that it is a pentagon. That's going to be how we identify this flight of family. So, let's go ahead and start with our rough stink bug here. And if you would like to ask any questions about the specimens, feel free to ask. The specimens do exist. If you did really want to look at them underneath a microscope or to get confirmation on a different character, I would have to go and grab the specimens from my collection, but we can totally do that if we want. So in stink bugs, some of the stink bug characters that are specific to this species are the jugum, or not the species, but the family. Stink bugs in general, Pentatomidae, have a jugum and a tylus. The jugum are these outside pieces, And the tilus is the internal one. And if you look at the very edge, identifying characteristics for when you're like looking at the keys and trying to identify stink bugs, there will be a lot of questions about the jugum and the tilus, how long they are in relation to each other, if they are even across. There's all types of characters there. This is great, like a review of the ones I didn't get to see live. Perfect! That makes me happy. And because I also sketch with students, there is definitely a likelihood that we're gonna sketch some of these stink bugs that, I actually didn't do live. Some of these I might have sketched with students. When I was first starting this sketching, I definitely wrote in the bottom corner kind of when I did it, because I was keeping track of who had seen what. But then it just got to be too much. It got out of control. There are too many bug sketches in my life. Not too many, but there are so many that I actually lost track of which one went where. So we've got our head kind of figured out. We're going to add these antennae. So stink bugs are going to have straight antenna. We also call straight antenna filiform. You may have heard that term from me before. The head is kind of connected into the pronotum here. I'm going to go ahead and give us this rough edge. So I purposely just made it kind of a little jagged here. To give it just enough texture to make it pop. Yay! Alright, so this segment right here, that's the pronotum. That's the first segment of the thorax and where our first leg is connected. Now, my stink bug legs are walking legs. They don't have any specialized kind of, I gotta turn this light on. There we go. Then the shadow's not so dark. They don't have any type of, like, specialized legs. They're not running legs or swimming legs. They're just all, 3 pair of walking legs. So we've got little tarsal claws that reach past the antenna, and that's fine. Alright, so our body is going to be moving down just a little bit. This whole segment in here on stink bugs is called the scutellum. That is spelled, since I'm sketching, I'm not like writing as regularly as I normally do on the thing. So I'll spell it to you in person. If you wanted to spell this word, scutellum, that is S-C-U-T-E-L-L-U-M. And that's this part right here. It's generally in between 2 pairs of wings. So you can see that here. Stink bug wings, because if we look at the name stink bug, I also have this. I forgot to put that up. If you also look at the name stink bugs are 2 words, which means that this is actually a true bug. It is an insect, but it is also an insect that has a piercing and sucking mouth part and what we call hemielytra. So if you recognize the word elytra, it's because we've talked a lot about elytra when we are going over beetles. Elytra are those hard outer shell wings that kind of protect the underwings of beetles in particular. So like a ladybug has elytra, and these are hardened shells that are their first pair of wings essentially, and they protect the membranous wings underneath. Well, true bugs have hemielytra, meaning they're half leathery and half membranous. And I always just kind of add these lines in here down here to kind of show that there are membranes down here. There are veins in this area, but those veins, they are really hard to kind of get all of the exacts down and are generally not used for identification. And that's going to be my rule of thumb, is if it's going to be used for identification, I want it in my sketch. But if it's something like these guys, I might just do them kind of these swirled lines. It gives it the same texture without spending a lot of time on something that you may not use. All right, and then if you look over to the edges, those are the abdominal segments. So in my rough stink bugs' abdomens are actually wider than their wings. So you end up seeing a good number of their segments along the edges. And then they have this darker, it almost looks like they have this banding on their abdomen. And if you look at the individual segments, each one of the bands, they have 2 bands per segment, and there's one on the top and one on the bottom. So you can see these darker bands along. All right, let's get my legs taken care of. I do remember almost running out of time when I was sketching this friend here, which is why I put the middle leg underneath so we didn't have to worry about those tarsal segments. But the hind legs are really difficult to hide in stink bugs, so the hind leg got to come out. Oops. Trying to keep my lines even. There's a part of me that wonders if I should be taking these pages out of the journal to do this, because I'm left-handed and every now and again this gets in the way. But, alas, we're good. Is the hemielytra used for flying or just held out of the way like a beetle? The hemielytra are mostly just used as a protection like a beetle, so they just kind of open them up and hold them out of the way. Good question. Hemielytra. Exactly. And so that's hemi. So you got it. Hemi is the Latin root for half. I don't know if it's Latin or Greek. Hemi is the root for half, just like a hemisphere. And hemiptera, P-T-E-R-A, ptera, that is rooted in the word wing. And so when you say hemiptera, what you're literally saying is half wings. And the whole order was named after the fact that their wings are half dark and half membranous. And I'm going to come back in and I will add these. All right. And one of the Exactly. Lepidoptera, scaly wing. Diptera, 2 wings. Hemiptera, half wings. Let's see. Dermaptera are the earwigs. And derma means, I believe derma means skin. Yeah. So derma means skin, and dermapterans, or the earwigs, are literally named kind of skin wings, which I've never really understood too much because their wings are elytra, just like beetles would be, but their wings are kind of halfsies. Pachyderm. Yep, skin wings. Yep. Pachyderm. Thick skin. Neuroptera. Yep. So you got it. Exactly. Oh, you know what? I never really thought of the word ner as, like net, but that makes sense, for, like, neurons in your brain, because they're kind of like a whole net that kind of connect together. That's kind of cool. Alright, so when I'm closing in, I want to make sure I crosshatch in the eye. Alright, so I went ahead and I crosshatched in the eye, and now the moment of, my eraser is green, so you can see through me. But now, for the moment of truth, I'm going to go back in and erase all this pencil to see what our line drawing looks like. I love erasing the pencil and looking at how clean the sketches are afterwards. There's a part of me... I'm trying to figure out if I should do these darker lines on the abdomen. I kind of want to leave those in pencil. But I probably shouldn't. I should probably just go in and... What I'm going to do is I'm going to come into these and I'm just going to outline the outside of the stripes. So that when people... So, you know that, I think I've mentioned this a couple of times, you know that eventually, once these sketches are line sketches, I'm hoping to turn them into a coloring book. And so, that's why I've been trying to keep kind of the coloring and the shading to a minimum, so that people can kind of color and shade it themselves. But if I give them the outline of the stripes, then they'll know that they exist. And they can feel free to color the stripes whatever color they want. So, what do we think? think that's how. Yeah, so I definitely need more thicknesses of pens. This is the only one I have. Or go over the lines that outline the abdominal lines to make those ones thicker. You're right, to make sure that they kind of stay separate. So it makes me happy that you use microns too. I use microns mostly because they are archival ink and they're a good pen for when you are using a pen to write your insect labels, because insect labels need to be durable from ink, not ink. They need to be durable from like water and sunlight, and you want your labels to last. And so I use a micron pen because it's archival ink, and if you write a label in archival ink or in with a micron pen, you can take that label and drop it directly into an ethanol vial, and it will not and it doesn't dissolve. It doesn't go away. It stays put. So when you're making insect labels, your choices are pencils or archival ink. And microns are my favorite. This one's a 0.3 micron. Yes, exactly. Some people don't realize that because all of us left-handed people, our hands kind of drag in the ink as we're sketching along. It's really difficult to get a pen that doesn't smudge when you're writing. And so, super happy about them. Platinum carbon black ink in a fountain pen. I've actually never used a fountain pen before, so I would definitely have to use it kind of as an experiment. Yeah, exactly. Alrighty, so I think our friend here is pretty good. We've got, I think I like the rough side over here on the left more than the right. I think this tooth is just a little bit too sharp. But those are the types of things that we learn when we're going through this. Here we go. Actually, there's a part of me that really wants to jump over to the soldier fly. Let's do the soldier fly. Jack says, regularly irregular. I like that. Kind of like, you want to try and make things natural looking too. And exactly straight lines don't happen so regularly out there in the world. Yeah, the soldier fly is a pretty awesome insect. You know, I actually might still have... The soldier fly was recent, so I might still have the soldier fly hanging out right behind my green screen. There it is. sneak peek at my books. So this little friend here is the soldier fly. He's all in yellow. And I have to turn on my microscope because I didn't turn it on today. But that is fine. Let's see. We're going to switch over to this one. Oh no! I'm too tall. There. Alright, so this is our friend, the soldier fly. The soldier fly has a handful of really cool parts on its body. We can make that screen even bigger. Zoom. That works for me. Alright, so this is our soldier fly here. Some key characteristics that we're going to be seeing, especially in our line drawing. Obviously, having really cool looking antenna is one of those things that we like to see. These antenna, there we go. Oh, come back. Alright, so these antennae are what we call aristate antenna, and they are these are kind of the generic fly antenna. They have 3 segments, and then they have this single kind of hair coming off of the antenna. This single hair here, its name is the arista, spelled A-R-I-S-T-A. And the entire antenna name are the aristate antenna, named for this beautiful hair that sticks off of it. Yeah, my scenery reveals a bookshelf, and it is definitely all back there. It's definitely full of bug books. I have most of my books that people might ask questions about, like my identification books, right here on this side of the scenery. But back there, I have all of kind of the special ones, and I have a handful of like old textbooks that have older knowledge, but I can't bring myself to get rid of them because I love reading about insects, even if it's old knowledge. Plus, I've got some really cool wing venation guides and stuff. Alright, so this is the other super interesting thing about our friend here. Those are halteres, and we can see them super easy. H-A-L-T-E-R-E. And these are the modifications of the hind wing. Flies are dipterans, 2 wings, which means, and they are actually one of the only insects that have 2 wings, the flies. Flies have 2 wings. And a lot of times the halteres are a little difficult to see because they're kind of tucked underneath the front pair of wings. But our friend here decided to open up his wings a little bit and show off his halteres. Aristocratic antenna? Do they go to the classiest parties? Yes, that is the answer. Our fly is an aristocrat, and it only drinks tea with its last tarsal segment out. Alright, so now that we've gotten a chance to see our friend over there on the microscope, we are going to come back here. We are going to inkline it. Outline? Inkline? What do you call what we're doing? Is there a name for what we're doing? I would like to say finalizing our sketches. That almost feels right. I just do it because I like bugs. That's all. I'm still very much in the learning process for, like, on the art side of things. I know that many of you are out there and are beautiful artists. My approach to sketching is a little more scientific, is like on the more scientific side of things. I try to make sure that everything is kind of proportionally correct between all of the body parts, and then just making sure that the shapes are about right. And so for me, when I'm sketching, I am a lot of times not looking how I can add texture or add flair to my sketch, but keep it pretty basic and correct with the anatomy, the anatomical features of our friend here. Now, you can see our little 3 circles here. Those are our ocelli, spelled O-C-E-L-L-I. And they are simple eyes. Those guys right there are in addition to our huge compound eyes. Alright, so that's one antenna. So our antenna has one stalk. A second segment here that kind of is a holder, a placeholder for this third one that was kind of open and slanted on the side. And then, oh no, that was not a very great arista. The arista. This one decided to come down here instead of up there. That's fine. Now, for me, going in and doing the cross-hatching on our compound eyes here is probably going to be a trick. I moved the edge of the pronotum down just a little bit. Let's go in and cross-hatch in these eyes. Now, flies have huge compound eyes, but that doesn't mean you get to crosshatch any larger. They just have many, many, many segments. So I'm going to see, trying to keep them mostly even. We've got one. I thought that I started it diagonally, and then it became straight, so admittedly, that's not my favorite. I might end up fixing that digitally. It's just that the eyes don't look like eyes without a little bit of cross-hatching inside of them. See, I wanted them to look both like this because our lines are diagonal this way. They create all of these X's. I think that when it looks like this and it's too straight, it ends up looking more like you've just accidentally just made a bunch of like tic-tac-toe boards. That's fine. Halteres are how I finally managed to figure out my obscure ant mimic picture winged fly. Oh, that's cool. Yeah, so those halteres kind of helped you identify that. Sorry, I hadn't looked at the chat for a minute, so let me go ahead and catch up. I really thought it was a winged ant, but it was funny looking. Yes! Yeah, has halteres means it's a fly. But I think that for sure, it makes me so happy that people are going ahead and using what they've learned, to help them identify, and they're using kind of the knowledge that you're gaining in real world situations or out in the real world when you're identifying or sketching live journaling and stuff like that. So, I want to say thank you for sharing that, because that makes me very happy. You got to see some of Nabokov's butterfly sketches recently. Nabokov's. I'm going to have to look it up. Oh, look at that. Well, thank you. Wow, he is a beautiful... I don't know. He's got something on me. The shadings on those butterflies is absolutely gorgeous that I'm looking at a handful of his sketches. But thank you. I super appreciate it. Oh, the genitalia. Okay, fair enough. That makes a lot of sense. So, when I'm sketching, I'll go ahead and pull out a book for you. This is The Aquatic Insects of North America. And when I'm sketching, a lot of times I'm thinking of pictures like this. These actually have a little bit more features of the individual hairs because they're talking about the hair length in these images. But when I'm drawing, a lot of times I'm thinking, like, I'm sketching mostly the outlines for kind of like this, for examples in our books. And then one day I'll move over to something like this where I'll start using the little dots. I have not gotten used to, we call that stippling. I've not gotten used to stippling just yet. But look at how cool this is. And these are just the line sketches. And so that's kind of when I'm sketching and thinking about what I'm doing. A lot of times I think about some of the sketches that I've seen in this book. Because I find that book has, when you're going through the keys, the pictures are so incredibly helpful. Especially when sometimes insect keys get really wordy. And sometimes it's hard to understand exactly what the author is trying to say. And so having just a sketch or an ink line drawing of what they're talking about is super helpful. And so I believe that these had these kind of caps on the end of them, just like the antenna did. And actually, when I first started sketching like this, it was because I was trying to figure out a fly's wing. In fact, I think I still have that sketch in my adults book. I believe this unknown fly wing was one of my first kind of attempted insect sketches, because I was so confused by this fly that I went over and over and over and over it and couldn't figure it out. And finally, I believe that I figured out that it was a canopid. I don't remember the common name for those. Sorry, guys. You don't think that the direction of the cross-hatching is too important? So do you like both of those? Because to me, personally, I much prefer the one on the left to the one on the right. But if you say that it doesn't matter much, then maybe that's okay. Oh, he wrote the book Lolita. All right. I've actually read that book. I forgot he was the author. Ooh, Jack Laws has some videos on stippling. See, that I'm definitely gonna need to get into. And I need to spend a little bit of time practicing essentially off camera. Or maybe with kids. Maybe I'll practice stippling with children first. So with our abdomen here, I will a lot of times, especially with an abdomen that kind of gets wider in the middle and then gets smaller at the end. There is a method for, you want to pick the widest and the largest part of the abdomen, and you kind of put a point there. That's kind of your visual, where you're kind of looking at it from. And then all of the lines above it are going to arch up, and all the lines above it arch down. And so what that creates is this shaping on the abdomen. Where you can obviously see that this is the largest one, and all of the other segments either look like they're coming towards you or going away from you. And our little fly here, I believe it did have these two little cerci. So it has these little appendages at the very end of its abdomen. They're two segmented. They kind of look like that. All right, we're going to do the wings, and then we'll go over here and we'll do the, I said wings. We're going to do the legs, and then we'll come over here and do the wing last. All right. A useful sketch for this purpose is one that makes the shapes and proportions really clear. Yeah. Well, and I guess that's also a thing, is making sure that shading does help with 3D structures. So it kind of helps understand, like, depth. It helps you with depth. So even though I'm not super shading it, you know, I might even, this area right here on the top of the abdomen is supposed to be kind of shaded and dark because it's deep. It's further away. So I just dotted it in just a little bit. We'll see what it looks like when we're erasing our pencil. Because we want to make sure that the abdomen looks connected, not just by this point here, but it does have a pretty wide connection in this fly. It's just that this is kind of above and that's below. So, good point, for sure. Having shading helps all those things. I love looking at wing venation. Yes. You know what? Doing butterflies like that with where you do the wing venations first and then the colors based on the wing venation or around the wing venation is a really, really smart way to do it. And the butterflies, if you are already getting practice in kind of looking at butterfly wing venation, underneath the scales of the butterfly's wings, then you're already actually pretty ahead of the curve, because those butterfly wing venations can be really hard to spot. Especially when you're trying to see them kind of through the scales. And the wing venations on butterflies is pretty much the only way to identify them to family. I mean, there are a handful of special cases like the lycaenids, the little blues. They have black and white striped antennae, so that's really easy to tell apart from everyone else. But most other butterflies and moths, they have to be identified by their wing venations. And sometimes that's really hard to see. So I'm trying to figure out exactly what I was sketching here, because I believe that this is the start of the wing. And the leg came out right at the same exact place. And I mean, that is the true case for this fly. And technically this wing is straight, so this line would just continue straight. But I don't want them to be the same line. We're going to do the leg first, and then we'll see about fixing that wing. All right, so, the wing venations on butterflies. The wing venations on butterflies, they're not all the same, but structurally they are very, very similar. So if you're looking, I'll have to get you a better sketch. I'll show you in a minute. The front wing is going to have kind of a cell on it. If you've seen that kind of long, wide cell, then at the end of the cell, there can be either 2, 3, or 4, I think, veins coming off of it, and you have to know how many of those veins happen. For moths like the swift moth, for moths like the swift moth, they will actually have the exact same wing venation on the front and the hind wing, whereas most other insects have unique veins on the front and the hind wing. So my foot ended up going just a little bit left, but I wanted to keep the pen lines even, so our foot just strayed just a little bit. And I feel like sometimes that happens in sketches. Now, I'm going to take this, I'm going to erase the pencil happening here. and I'm going to fix this. So what I think I'm going to do is I'm going to take where this wing would be, and I'm going to kind of point it down rather than pointing it out. I might just leave it without the wing veins. I don't know. I like this wing. I want to focus on this wing. So maybe I'll add... I think what'll make this look good is if I add the wing veins. So 1... I believe there's another one. All right, let's get this one done. Yeah, so I think that when we were sketching these veins, I actually went over all of the names of all of the veins. And we talked about wing venation in general. Now, after I get this sketched, I want to show you a really cool old book I have that has all types of wing venations in it because you mentioned that you liked them, and I want to share with you a really cool book I have. Oh, he's so cool! Yeah, and so you are starting to notice some differences, especially the differences between a swallowtail and monarch butterfly wing venations. That's definitely something that, if you look really closely, you'll be able to tell different families of butterflies apart by their wings. Yeah, exactly. All right, so I'm glad that we like it. So I think we've got all of our lines figured out. I'm so excited to erase this pencil and look at what it looks like underneath. And I believe this is, I don't believe we're going to be doing another one, but I do want to pull out the wing venation book for you so that we can look at it together. I believe it's also on the other side of my magic wall. So I'm gonna disappear for a minute. My eraser says, "Oh no!" This is what you're used for, Mr. Eraser. So that is our soldier fly, the Stratiomyiid. Cool. Morning, Chaos. Alrighty, so I have this book here. Its publishing was 1906. It has stamps from the MSU Entomological Department from January of 1907. And interesting thing about books from this long ago is that sometimes the pages aren't all the way trimmed, but I was hoping we might flip over to the other one I pulled. There's two of them. So we've got very distinctive sketches in this book here, which is kind of fun. But the other thing I wanted to show you is that this book does this, which is unique. I haven't seen it in any other book. It does these dark and light patterns inside of the wings to show how the wings fold over the body. And so you can see something like this where the wing veins are kind of folding this top in, or kind of folding the edges over, or this one where they're kind of all folding in. This is the wing of a staph linnet or a rove beetle, so it has to fold very, very, very small to fit underneath the really small elytra. So I thought that that is a really cool way to look at the wings, and I wanted to show you that. And it is night for us. Yes, it is almost 11. And this is the guide to the study of wings of insects. And this one is one of my favorites. This one is copyrighted in the 1930s. And so the 1930s is when entomology, in my opinion, was like at an amazing point in its life, because in the 1930s, when people were going and studying insects, they were studying insects because they loved them. They were publishing papers because they were learning all of these really cool new things. There's a lot of identification guides, a lot of life history guides, a lot of entomologists that were going outside collecting native insects and just rearing them inside because they wanted to see what they would eat or how long they live. And those types of very baseline science, that's what strikes my chords. That's what makes me happy. A lot of times entomology now, or like a lot of times the research or the scientific research nowadays has to have something about the bigger picture. Whereas back in the 1930s, they were just going out to learn. All right. And this book has this. This is a wing vein, and it has every single one of the veins labeled and numbered along with all of the cells. And there are, I don't know if I've ever even looked through every single one of these plates. There are something like, oh, look at that one's just a little one. Those are little wings. Oh, that's a Hymenopteron. That's a Vespid. That's a paper wasp wing. Whoa, this one's cool. Oh, this one's from a fossil. This is a fossilized, this is the Permian fossil dragonfly's wing, Venation. I know you can't get grants on the fact that just insects are cool anymore. Or, you know, and so many people were self-funded back in the day, and it's so hard to self-fund and publish your own research, and it just is just a rough one. So, yes, and so there are all of these plates, and every single one of these plates is drawn and labeled and numbered, and they look like they are hand sketched by somebody, like somebody went and copied every single one of the plates on kind of a larger piece of paper, but that's pretty cool. So this makes me happy. I wanted to show you these. And I thought that you would also super appreciate seeing some of these wing venations. Yeah, so here's the thing. The entomology department at MSU, when I was there, they were going through their library and they were getting rid of books. They were throwing them away. And so when they had a bunch of doubles and they were getting rid of all of their doubles. And so I ended up picking up a whole bunch. I ended up picking up a whole bunch of really old texts from, like, the early 1900s. I believe I even have some from the 1800s that were just being kind of tossed. The first one is called The Wing Veins of Insect by C.W. Woodworth. I thought it's called a griffin fly due to its differences. It possibly is called a griffin fly because of its differences. That's just what the plate said, and that plate was from the early 1930s. So that might have been what we called it back then, but maybe not what we call it nowadays. Oh, okay. So, you asked if the veins actually carry nutrients or if they are just for structural stability. And that's funny because I believe I was also asked this yesterday or 2 days ago. The answer is they do carry hemolymph for a very short time frame. So if you're thinking about a butterfly, and when the butterfly emerges from the chrysalis or cocoon, it's still kind of wet. When that butterfly is wet and when it's trying to expand its wings and blow out its wings, it's going to be pushing hemolymph through the veins, and it's going to push and expand those wings using the veins by pushing their blood through it. But once the wings dry, they actually close off the connection of hemolymph to the veins in the wings. So once the wings are dry, there is no longer nutrients or blood flow through the wings, and that's why if the wings get damaged, an insect doesn't bleed out. Oh, yes, it is hemolymph, spelled H-E-M-O-M-M-O-M-P-H, hemolymph. So humans have hemoglobin. That's what we have, that's our blood, and it does have the ability to transport oxygen. Hemolymph is what insects have, and their blood does not transport oxygen. Insects have a completely different respiration system where they breathe through diffusion through their spiracles, and then each one of their, the spiracles go into something that we call tracheoles, kind of like our trachea, but it's oles because they're tinier. The little tracheoles that split and split and split, and they feed every single cell in the insect's body. Something that I have never taken is insect physiology, and that's something that I definitely would like to learn more about. I do know the basics of how the internal workings of insects work, but but I would love to get into their physiology and how all of their little parts actually work. I think that that's pretty cool. Hemo is iron, like the mineral hematite. Ooh, I didn't know that. That's really cool. See, we can learn from each other. That makes me happy. All right, so those are the 2 books, The Wing Veins of Insects and The Guide to the Study of Insects by Bradley. And they're 2 of my favorite books for wings. And this one, admittedly, I haven't read all the way through. I sometimes am afraid of opening some of these books, but also, look at this. So these are 2 separate pages, but they're connected at the top. And this is something that happened, I guess, frequently. And so I don't know how people fixed this. You know, I don't want to just rip these pages, but it was from, I'm guessing it's from the printer press, not cutting it in the exact right place. And so many of these pages I actually can't turn because they're connected at the top. And some of them are even connected on this side too. Like there's a page in here that's connected. You can see it's starting to open here. And so, if anyone knows more about books and the safest way to go about opening this book and reading it fully, I would love to know. Do insects have a heart? I will go ahead and draw for you, ladies and gentlemen, the internal workings of an insect as I know them. We're going to use this page here. It's the back side of something else that shouldn't matter too much. Now, I'm going to draw us a grasshopper because a lot of times grasshoppers are, funny enough, they're kind of what we use as a guide for like body parts. A lot of times when you're learning the body parts or the internal workings of an insect, you learn at first about grasshoppers for some reason. So if we are talking about grasshoppers and their internal workings, we asked specifically about the heart. Now, insects have what we call an open circulatory system. So, they do not have veins per se inside of their body that moves their blood. Alright? They mostly, their blood just kind of floats around in their body, and their organs just kind of float around in their blood. But, they do have what we call an aorta vein. Alright, it runs along the top of the body, and essentially it's just one long tube with individual little pumps. And these pumps, they pick up the hemolymph from the back end of the body and spit it out at the front of the body. And that is what creates the circulation of blood in an insect's body. So they call this part the aorta vein. And is the closest that you're going to get to a heart in insects. They also do have a nervous system of sorts. They have what we call ganglion. They've got these kind of nervous systems. It runs along the bottom side of the insect. And then every so often you have kind of a cluster of nerve cells. And that is what helps, and that is the insect's nervous system. Now, they do sometimes, people will say that insects' brain, and regularly they're going to have a larger cluster of ganglion here over near the head, and they can generally consider that kind of the brain. But keep in mind, I haven't taken insect physio, so this is just how I understand it. Then, if you want to get into the stomach, that gets kind of complicated. I do have it written up if we are interested, but it's like, the insect eats the food, it goes into a storage area that we call the crop. It gets pushed through the ventriculus, which is a really kind of small opening that starts kind of help grinding the food into smaller pieces. And then it goes into the midgut, and the midgut is where the digestion process starts. It's equivalent to kind of our stomach. It's where the enzymes are that start breaking stuff down. Then if we go a little bit further, they're going to have these weird things. What are they called? They're like fingers in the body. I don't remember what they're called, but there are these like little fingers that come off of the system that kind of help clean the blood. So every now and again you get waste that ends up kind of floating around in the hemolymph. And so insects have these little finger-like projections off of their kind of midgut that will clean their blood and pull all of those bad things back and put them into the, essentially, when you're moving into the hindgut, this is kind of like the insect's colon. It helps kind of squeeze and get all of those nutrients out, and goes out the end. There are also, I believe, what we call rectal pads back here at the right before the exit, and those are kind of like these spongy pads that even kind of sponge the pellet dry, or sponge even more of the water out of it, because the insect's goal is to keep as much of the water as possible. Dend something. Let me look it up. They are called Mel Pigian Tubules. I never remember the name of those. And those are the little finger-like projections that help clean the insect's blood. All right, so let's see. So I got the do insects have a heart from Chaos. Wait, Chaos, you're a linguist. Can you weigh in? We've been discussing Latin roots. Yes. Traditionally in bookbinding, many pages were printed onto a very large sheet of paper, which is then folded a bunch of times. Exactly what I'm seeing here. Yeah, so it's like there was one big piece of paper that they folded, and then they didn't cut it properly. Ah, yes. Okay, that makes sense. So I wonder if maybe I could use kind of like a letter opener to individually separate the pages. All right, so that is our day today, ladies and gentlemen. I wanted to get a couple of really fun sketches in. So we got our, let's see, we got our Mina, the rough stink bug, in ink and done. I'm pretty excited about that one. We skipped the blister beetle, but that's okay. We'll come back to it. And we did the soldier fly. And we got talking about wing venations, because wing venations are super cool. Alright, and that always makes me happy. Yeah, so you, some insects that you can see through, you're going to be able to see some of those things. And exactly, you may see this little pump that's pumping this blood. You may see the stomach. If you ever are looking in an insect abdomen and it looks like it's got like little strings or little veins all over the place, those are the Mel Pigian Tubules that are cleaning its blood. And we didn't talk about butts. Look at us, we win or we lose, depending on if we like talking about butts. Let's see. Oh, another cool thing about caterpillars is if you look at them really, really closely and you watch them move, somebody put a caterpillar in front of essentially an X-ray machine and was watching the way that it moves. Caterpillars will actually throw all of their internal organs forward first, and then their legs follow. So instead of actually using their prolegs to kind of walk and move, because their prolegs are just muscles on their abdomen, they actually use their internal organs to move. They just kind of throw their internal organs forward and then their legs follow. And that I kind of love because it feels so opposite to what you would expect. We love talking about bug butts. Darn it. So, maybe next time we'll have to spend a little bit more time talking about bug butts. Alright. Ooh, thank you, Chaos, for the bonus information about linguistics. About the English language. That's fascinating. Alright. So, I do want to say thank you so much for joining me tonight. I do believe the pen live streams I'm going to try to keep closer to an hour, just so that by the end of the month we aren't all the way burned out. We want to still be having fun and being upbeat and interactive. And so, we're going to be closing out for the day. Keep in mind, I'll be back tomorrow, so you won't have to miss me for too long. Tomorrow is Thursday, so we will be doing a brand new pencil sketch at 10 p.m. Eastern. I look forward to seeing all of you there, and I need to jump over to my closer so that you guys get to see all of the bonus things. Over there at the top, it's a green paintbrush. I forgot. Over there at the top, is Out School. I believe you all have heard this spiel before, but I'm going to say it again. That is where I teach online classes to students ages 5 to 8, 9 to 12. I can do high school classes. I don't have any high school students just yet, but hey, if you know a high schooler that wants to learn more about bugs, I can teach them. So, this is your reminder to subscribe to the channel if you're not already subscribed. Thank you so much to those of you who are and who have those notifications turned on so that when I go live, your phone goes, buzz, buzz, Trisha's actively chatting about bugs, come and join her, right? So this over here is my QR code to my PayPal account. It is just to say thank you. If you have a, if you've really enjoyed today, if you've learned something, if you want to buy me a coffee, you can go ahead and send me a couple of dollars through PayPal. That is all donations to Insectopia to help me purchase things like new sketch pads and more pins and collecting gear to keep our sketches and to keep everything upbeat and interesting. We're keeping it all fresh and new. Super appreciate anybody who in the past or in the future who have donated to this cause. It means the world to me. Absolutely. Now, if you are looking for me on social media, I'm at Insectopia 2015, not just at Insectopia. Unfortunately, somebody else took those. So, at Insectopia 2015 on both Facebook and Instagram. But I believe on TikTok, I'm just at Insectopia. I don't have very many videos posted, though, so that's something on social media that I should work on a little bit. All right. So, thank you so much, ladies and gentlemen, for spending your Wednesday night or Wednesday morning with me, Chaos. Thank you for spending your Wednesday with me, and I will see a number of you tomorrow for our Thursday night live streams. Those are very well attended. So, thank you so much, ladies and gentlemen, and I will see you tomorrow. Stay buggy!